Good morning. I am Professor Sunil Khare from Indian Institute of Technology. We are working on e-Pathshala program of UGC, for which I am coordinator for the subject biochemistry. Under this, we are discussing a paper on structure and function of biomolecules 2. The today's module will cover the basic components of DNA, which means the sugar present in it, the phosphate present in it, and the bases present in it. We will try to understand about it. In today's lecture, we will explore the historical perspective which led to the discovery of DNA or nucleic acid as genetic material. This will involve looking at some of the classical historical experiments and also the gradual timeline of discovery of genetic material. The concept map for this module is, if we look into nucleic acid, we will first briefly study what are nucleic acid, how these were discovered, what is their function, and how many types of nucleic acids are there. Then we will try to understand the structure of nucleic acid in detail and look at each of their components like bases, sugar and phosphates. We will see what are nucleosides, nucleotides, and finally, we would like to understand how polynucleotides are formed. The nucleic acid constitute the most important biomolecules of the cell, and these are critical entities for all forms of life. Discovery parts should be understood as nucleic acid were first discovered by Friedrich Mischer in 1869. He reported that he has found a substance within the nuclei of human white blood cells, which was weakly acidic in nature and the function was unknown. He had named this material as nuclein. Few years later, Mischer was successfully able to separate nuclein into protein and nucleic acid components. Nuclein was later named as nucleic acid in 1889 by Richard Altman. They were so named because of the initial discovery from within the nucleus and due to the presence of phosphate group in their molecules. The function: nucleic acid are present in all living beings as well as in bacteria, archaea, mitochondria, chloroplast, virus and viroids. Nucleic acids are involved in the storage and transfer of genetic information in living organisms. There are two types of nucleic acid in the cells: the deoxyribonucleic acid DNA and ribonucleic acid the RNA. Both DNA and RNA are the molecular repositories of genetic data. The basic components of nucleic acid include three different entities. As you see in the figure, a nitrogenous base, a sugar moiety and a phosphate group. These combine to give one unit of nucleotide, and these nucleotides are stacked in nucleic acid molecule. Now, first coming to nitrogenous bases. The nitrogenous bases are nitrogen containing bases which are derivatives of two heterocyclic compounds, purine and pyrimidine. Pyrimidines are monocyclic, whereas purines are bicyclic. These bases are polyfunctional in nature. Purine bases are composed of 9-membered double ring structure with 4 nitrogens and 5 carbons, while pyrimidine bases are composed of 6-membered ring with 2 nitrogens and 4 carbons. The carbon and nitrogen atoms in purine and pyrimidines are numbered based on the convention which is shown in figure. The basic structure of purine and pyrimidines remains same with some modification. The nitrogenous bases inside the cell, 5 major nitrogenous bases are found. The derivatives of purine are called adenine and guanine, A or G, while the derivative of pyrimidines are called thymine, T, cytosine, C, and uracil, U. The DNA contains A, G, C, and T, whereas RNA contains A, G, C, and U bases. The principal structure of these bases is shown in the figure which you can see. Now, let us look at the properties of purines and pyrimidines. Both purines and pyrimidines exhibit tautomerism, that means they exist in keto and enol forms. The keto tautomer is known as lactam ring, whereas enol tautomer is known as lactim ring. At neutral pH, the keto tautomer remains the more predominant. Upon interaction with other molecules, ring nitrogen in the lactam serves as donor of hydrogen bond, and the keto oxygen behaves as hydrogen bond acceptor. Very interesting property of DNA is, as a consequence of aromatic ring structure and associated resonance, pyrimidine and purine bases absorb ultraviolet light or UV light with an absorption maxima at a wavelength 260 nanometer as shown in the figure. The measurement of the concentration of DNA or RNA in a given sample is therefore performed by measuring the UV absorbance at this wavelength. Purines and pyrimidines, being complementary bases, can participate in base pairing. Based on the specific shapes and hydrogen bond properties, the base pairing occurs. Guanidine, being a complementary to cytosine, pair with cytosine through 3 hydrogen bonds. Adenine is component of thymine. It pairs with thymine with 2 double bonds. Same adenine in case of RNA will pair with uracil. The pairing of these bases are shown in the figure which you can see. Here is very important aspect of DNA. Very early studies emerged from the laboratory of Chargaff. Erwin Chargaff, an Australian American biochemist, gave the Chargaff's rule, according to which DNA always contain equal amount of certain base pairs. he observed that amount of adenine will always be equal to amount of thymine, whereas the amount of guanidine will always be equal to the amount of cytosine, regardless of the source of DNA. The ratio of AT to GC varied from 2.7 to 0.35 in various organisms. Two types of pentose sugars are found in nucleic acid, namely ribose and 2-deoxyribose. The carbons in the ribose sugars are numbered according to convention shown in the figure. The ribose differs from deoxyribose in the presence of hydroxyl group at 2 position. The structure of both ribose and deoxyribose are shown in the figure. The D-ribose and deoxyribose are found in RNA and DNA respectively. These are found in fumarose forms. Phosphate is another important component of nucleic acid molecules. It gets attached to C'5 hydroxyl group of the sugar and gets incorporated into nucleic acid, both DNA and RNA. A nucleoside consists of a combination of nitrogenous base and a sugar, either ribose or deoxyribose. So, this means nucleosides can be written as nitrogenous base plus sugar. The bond between them is called beta glycosidic bond. Position of attachment can be seen in the figure. The example of nucleosides would include cytidine, uridine, adenosine, guanosine and thymidine, as you see in the figure. Nucleotides are comprised of a nitrogenous base linked to 5 carbon sugar and one or more phosphate group. The phosphate group is attached to 5' hydroxyl group of sugar part of nucleoside. They function as building block of the nucleic acid. For making it easy to understand, you can say if a phosphate is added to nucleoside, it will become nucleotide. So, the nucleotide can be written as nitrogenous base plus sugar plus phosphate or nucleoside plus phosphate. Formation of nucleotide is described as the base of nucleotide position N1 of pyrimidine or N9 of purine forms a covalent N-glycosyl bond with first carbon of pentose by removal of a water molecule. The phosphate is esterified at 5' carbon. In the picture, you can see the formation of nucleotide. Therefore, the examples of nucleotide would include deoxyadenosine monophosphate, deoxycytidine monophosphate, deoxyguanoside monophosphate, deoxythymidine monophosphate. All these 4 are shown in the figure, which you can see. Nucleotide di and triphosphates are the term nucleotide generally refers to a nucleoside monophosphate, but in case additional phosphoric acids are present, they link to the existing phosphate, they can produce nucleotide diphosphate or triphosphate, which is very clearly shown in the figure. Nucleoside monophosphate can sometimes form 2 ester bonds with phosphoric acid at 5' end and 3' hydroxyl groups of the ribose sugar. This therefore results in the formation of a cyclic nucleotide. These are aberrations, but abbreviated as CNMPs, where C stands for cyclic and N stands for respective nucleotide. The figure shows a cyclic guanosine monophosphate, cyclic AMP, cyclic JMP. These are very important biomolecules of the cell, and you can see their structure in the figure. These play very important role in regulation of cell metabolism. Here is a table to give summary of nucleobase derivatives and their nomenclature. In this sense, this table summarizes whatever has been understood so far. For example, the nucleoside formed with adenine and deoxyribose sugar is called 2' dioxyadenosine, while that formed between adenine and ribose sugar will be called just adenosine. Again, the nucleoside formed with the adenine, dioxyribose sugar and phosphate will be called 2' deoxyadenosine 5' monophosphate DAMP, while that formed between adenine, ribose sugar and phosphate will be called adenosine monophosphate or AMP. Similar nomenclature is followed for other bases: cytosine, guanine, thymine and uracil. You can familiarize yourself with these nomenclatures from the table. Now you see a polynucleotide. The polynucleotide formation is initiated when many nucleotides continue to join together by phosphodiester linkages. The formation of polynucleotide is catalyzed by polymerase enzyme, the DNA polymerase in case of DNA or RNA polymerase in case of RNA. The OH group on the third carbon of sugar is one nucleotide reacts with phosphate attached to 5' carbon of another to form phosphate ester bond and a dinucleotide. Repeated formation of such bonds leads to further elongation of the polynucleotide chain, which can be seen in the figure. DNA and RNA are example of polynucleotides shown in the figure, where the nucleotides are arranged in a linear way and proceeds in 5' 3' direction. A common representation of polynucleotide is given as 5' PAP TPGPC hydroxyl 3'. You can see this in the slide itself. While RNA is a single stranded, DNA is double stranded and contain two such polynucleotide chains. These chains spiral around each other to form a double helical structure. The two chains in double helix are held together by hydrogen bonds by complementary base pairing on different chains. So, whatever we have learned so far in this module, we can summarize that nucleic acid are important cellular biomolecules. In fact, these are master molecules. There are two types of nucleic acid in the cells, namely DNA and RNA. Nucleic acid include three different entities, namely nitrogenous base, a sugar and a phosphate group. Bases are nitrogen containing molecules which are derivative of purine and pyrimidine. 5 major bases found in the cells are adenine, guanine, thymine, cytosine and uracil. Base pairing occurs between G and C and A and T. So, the Chargaff's rule says %A will be equal to %T, whereas %G will be equal to C. Ribose and deoxyribose are two types of pentose sugars found in nucleic acid. Nucleosides are nitrogenous bases plus sugar, whereas nucleotides are nucleosides plus phosphate. When nucleotides join together by phosphodiester linkages, a polynucleotide formation is initiated. So, in this lecture, we have understood the historical perspective of DNA. DNA is the master molecule or genetic molecule for each living cells. We also understood the different components of nucleic acid, namely sugar, phosphate and nitrogenous bases. We looked into nucleoside formation, the nucleotide formation and polynucleotide formation, which leads to the structure of DNA. The cyclic nucleotides were also discussed, which are important parts of the cell functioning. DNA is master molecule of the cell. In fact, both DNA and RNA are master molecules of the cells. The nucleic acid are thus very important molecule to study. The historical perspective which led to the discovery of DNA and RNA is very important. So is that what are the different organic moieties which make these molecules. In case of DNA, this is nitrogenous bases, deoxyribose sugar and phosphate, whereas in case of RNA, this is ribose sugar, nitrogenous bases and phosphate. The difference among the nitrogenous bases is, as you can understand, the RNA will have U or uracil in place of thymine. So, in case of DNA, it will be GCAT, whereas in case of RNA, it will be GCAU. Now, we have to look into the architecture, how these three components make this three-dimensional structure a master molecule of the life. You see, when a nitrogenous base is attached with the sugar, you will call it nucleoside. And when this nucleoside is phosphorylated, you will call it nucleotide. And many nucleotides, when they link each other by diester bond, this will be called polynucleotide, which is what is the very basic backbone of the DNA and RNA structure. DNA is double stranded and RNA is single stranded. What I would like you to understand, the structure and conventional nomenclature of nucleotide, that means their numbering of the carbon and nitrogen, and that of sugar, the numbering of the carbons, which will enable you to understand the structural architect. And in this case, the figures have already shown you how these are called by different names, the AMP, the GMP, the CMP and TMP.